right. So now we are moving to the next section of the chapter where we're looking at imaging and sort of stimulation techniques. So this will be scanning and recording for recording data about body, how it processes information, what the brain is doing, what activity in the brain looks like. So we're going to go over some of these recording and scanning techniques for the rest of this chapter. So one of the first scanning techniques is EEG, and ECG. This is electrocardiogram, and this measures heart rate as a measurement of arousal. So here it's you would put these little pieces sort of here on your chest, these little sort of sticker tags, and they would be able to measure aspects of your heart rate, just like simply from your pulse, and then as a factor of arousal. And then what it does is it captures heart rate variability, which is a measure that refers to the variation in time between beats. And the variation in time between beats and how the beats, how those beats are structured in terms of shape, and these other types of pieces of this wave, as you can see here, the PR interval, these different sort of particular components that here, this is sort of the QRS complex, and what does this sort of really point? Is it sort of wide? What does the recovery look like? Because the slope of some of these can change. And so you would have this measurement of this heart rate variability before each person. And the heart rate variability is basically a measurement of sort of the heart's ability sort of to like respond and to recover to a stressor. So you would look at sort of the beat and then its recovery time and how that would change from a stressful situation to a non-stressful situation. Right. The next is physiological recordings. And so this can refer to electrodermal activity. And this is a really fancy way of basically talking about the sweat on your hands. Electrodermal activity, also called skin conductance, is basically that when your skin gets a little bit damp, such as when you're sweaty and stuff like that, then it actually conducts electricity much, much better. And so you can actually have these measures that are on your fingertips that measure the skin perspiration, and this is then a measure of arousal and stress. So skin conductance, or the sort of amount of perspiration, is a measure then of arousal, and that is referred to as electrodermal activity. Then you also have electromyography, EMG, and this measures electricity put out by muscle activity. So, and this is often used on the face to quantify microaggressions and stuff like that. So these would be things that you might be here that put on your face, and they can detect tiny movements. And so if you're saying to quantify micro expressions, then it would be something like, and it can look for the muscles, in here in your face are very close to the surface. And so then it can measure the changes in these micro expressions, and you can actually tell a fair amount about a person's arousal and by those micro expressions on a face, and what aspects of their feeling or emotional states might be. And so electromyography compares some of these micro expressions and these muscle movements, and certain muscle movements tend to be activated in certain types of situations. So it can kind of compare and contrast these to also figure out a level of arousal. And all of these measurements have particular strengths in certain contexts, and then other contexts where it might be a little bit harder to implement. All right. So heading into some of the stuff that you guys might be a little bit more familiar with in terms of things that you run into in your own self-care. X-rays. You guys remember this stuff has been around for a long time. Part of the electromagnetic spectrum. X-rays pass through tissue, and it's through selective absorption. So the denser the tissue versus the lighter the tissue, there's differences in the absorption selectivity for the X-rays. And so when you put someone through, then this film behind you, that kind of, that's what the X-rays pass through and respond to the different densities in your tissue. And then whatever sort of comes out back on the other side is what, is detected and represented as an image. CT scans, they build from X-rays. So a CT scan is also known as a CAT scan, and it refers to computerized axial tomography. And this is basically when a series of X-rays arranged by a computer, in particular shapes, this X-ray beam passes through the brain at many different angles. And when it passes through at these different angles, it comes rise to all of these different images. And there's differences in tissue density for high density tissue, such as bone. This absorbs a lot of the radiation, so it looks very white. Whereas lower density materials, such as the ventricular fluid, absorbs very little. And so, and so then you can see that as a much darker color. Neural tissue absorption actually lies between these two extremes, and the density, for example, of white matter fibers, they tend to be much more dense, and so they're wider, while as gray matter cells and things like that, those are not quite as dense, and so they're kind of gray in between sort of the darker and the lighter colors. So the CT scanning software basically translates all of these different angles and absorption rates into a brain image in which dark colors indicate low density, and higher light colors indicate higher density. And because this is going through these angles and stuff like that, then it can also create more of a 3D image. Now, next is electroencephalography. So this is something that, is a lot of recording of basically sort of brainwaves through your scalp. And this, many people will have a ECG, electrocardiogram, in terms of measuring heart rate, and EEGs are also very, very common. You'll encounter them in a sleep study. So in EEGs, there's electrodes placed at different locations on the skull to detect electroactivity from action potentials in the cortex. And as you can see some of these here, this is just an example. This is a fairly older picture, but all of these electrodes are placed at specific places on the skull to detect certain signals that actually have meaning in terms of differences in electrical patterns and wavelengths. So EEG is advantageous because it has continuous recording, so it can look at brain waves for an extended period of time, as you can see, here. And you can also get event-related potentials. So these are brain waves that are time-locked to specific events like a stimulus or a motor movement. And so then what EEG does is it measures the summed graded potentials from many thousands of neurons. And then, because we have all these graded potentials, as we've talked about with the neuron, all of these have these sort of electrical, these electrical messages that are passing down the action potentials. All of that actually creates these sort of magnetic fields that go around, the neurons. And then there's these electrical fields, actually, I should say the electrical fields go around that also creates these magnetic waves. And so the EEG can basically sum these waves right, here, and to look at particular time point changes, and that to explore how EEG changes as behavior changes. And so an EEG recorded from the cortex can display in a range of patterns, and some of which almost appear rhythmical, which you can see right here. So this is where EEG is actually used the most. And As you can see over here, this is for a relaxed person, but this is someone who is awake or excited in panel A. In panel C, this is someone who is drowsy, so there's a slower frequency. And then this is deep sleep, so you have even slower and sort of like waves, here. I haven't been getting as much deep sleep lately, and I really would like to get more of it. And then you have B, this is actually relaxed, eyes closed, alpha rhythms generated. So this is just being in a relaxed state. You can see some of these pieces in B right here from having these alpha rhythms, and some of this is looking the same as you would see over here, which corresponds to this person being in a relaxing state. This is someone who's asleep, and you can see there's these shifts in terms of higher and lower amplitudes. And then this is someone, to just give you an idea of, this is someone who's in a coma. So you have very much like slowing of activity. And you can see here that as it moves along, there's very little of this intense sort of activity going along in these spaces. So it's just very slow and then not with many sort of facets to the wavelength that would indicate neuronal activity. So EEG is relatively inexpensive and it records in real time, but it can only get at the surface of the brain, so it cannot get into things that are real deep, which gives it this real limitation. But then again, it's particularly advantageous for measuring things like sleep. And can also sort of show just how a living brain is just like never silent. There's always sort of something going on. All right, so now we are moving on further into brain scans and imaging to MRI. So this is one of the most well-known imaging going on right now, and it's very, very omnipresent in psychological and cognitive neuroscience research to look at like what's going on in the brain in terms of behavior. And so the magnetic resonance imaging, also known as MRI, it uses a large magnetic field that line up the spin of protons in water molecules. So these magnetic fields are strong. They're safe, so they're tested and all that kind of stuff. They're completely safe. But what they do is they're strong enough that the water molecules in your body, when you put sort of a magnetic field, they'll tend to align with that magnetic field. And so usually our hydrogen atoms are sort of like bouncing all over the place. Here we are, moving in the water. And then when the magnetic comes through, they're like, oh, we're still dancing here. But they're now all in these sort of like vibrating within this sort of range within sort of within that aligns with sort of this magnetic field. So instead of sort of going all these directions, right and left, then they're just basically kind of pronating, spinning within this medical field. And so within this magnetic field, it aligns them. This is a portion of them. It's not like all of our molecules are forced in alignment, which might actually have. So this is a small portion of molecules, but you can still get these effects. So you don't need to have it with everything. And so what happens is that when the magnets are turned off, the protons sort of, go all over the place, and then they were responding to detectable radio waves and things like that. When they go on, then they start to pronate in sort of these specific directions and line with the magnetic field. So even if their normal direction might be like here, like, woohoo, we're over in this way, but when the magnetic field turns on, they go, whoop, okay, we're aligning in the magnetic field, turns off, okay, we're going this way, magnetic field turns on, oop, we're aligning again. And so then what you can happen is then this detects sort of these radio waves that are generated by the magnetic field and aspects of our sort of pronating and sort of spinning water molecules. And the MRI can actually detect some of these shifts in sort of how an alignment and how the hydrogen atoms are sort of flipping into these various stages. And it can use that to figure out what the tissue is in the brain. So for example, the different types of tissue have different types of density, and then the water molecules within that space, based on the density, then that can affect their movements in terms of sort of how free ranging they are. So high density might be sort of moving like, this, like low density can have much more sort of faster movements. And so all of the water molecules as a function of these different densities kind of move and align in specific ways. But then when you turn the magnet on, then they all try to align with the magnet. And when you turn the magnet off, they then spin back into their sort of own stations. Now, when they go back into their own movements, and then when they come in a line and when they go back, those changes and those shifts actually put off these detectable radio waves, and that's what the MRI machine can detect. So it's good for creating these very detailed images here. And so you can see the really nice differences in density. These are the white matter tracks. These are gray matter right in here. These are the ventricles that are filled with the cerebrospinal fluid. So you can see how these are really dark, and active brain tissue. Here's the skull. So it's sort of the widest, most dense piece of all. And this is just how they look. I've used to work with MRI. I did a fair amount of MRI work. I have a couple of publications on it. And this is actually exactly what the place looked like, where I'm wondering if this is actually a picture. This might be a picture of grad school from our imaging facility, because it literally looked exactly like this. But this is sort of what it looks like. You'd go inside and here, this is in this hollow tube. And so this is where it would align sort of these molecules within this space, and you would be able to detect these images. Now, what's special about MRI is that it also has techniques that sort of branch out from it that are also very highly effective. So MRI, when you can switch aspects of the protocol or switch aspects of the procedure, and you can use it to look at functional, like changes in the brain in fairly real time. So bold fMRI relies on bold contrast imaging, which refers to blood, blood oxygen level dependent. And this basically refers to, that when the activity in our brain, any of our neurons need oxygen. So we can look at the rates, the way the neurons consume oxygen to get an idea of how active they are. The more active they are, the more oxygen they consume. So if we look at how much oxygen they consume, can we reverse logic that to get an idea of like how much neuronal activity is going on within a particular neuron. So what this would mean is that then, as oxygen is being used from the blood, that the more oxygen that is extracted from the blood, then the more that area is using oxygen to get activities done. And when there's less oxygen, then there's less activity. So the MRI can detect these changes. The MRI scanner can detect changes in how the hemoglobin, which is what carries the oxygen, how the hemoglobin molecule changes, whether it's oxygenated or not. And so here we can walk through this a little bit. So active parts need more oxygen, as we discussed. Changes in oxygen content. The blood alters the magnetic properties of the water in the blood. So the hemoglobin, whether it has a water molecule attached to it or whether it does not, has different magnetic properties. So then we can kind of compare how much oxygen is attached to the hemoglobin, or how much oxygen has been removed from the hemoglobin. So oxygen is carried on this hemoglobin molecule in red blood cells now, and when we increase our activity, then this changes the ratio of the oxygen-rich hemoglobin to oxygen-poor hemoglobin, and this then alters the blood's magnetic properties. So This is because oxygen-rich hemoglobin is less magnetic than oxygen-poor hemoglobin. When the oxygen has left the hemoglobin, then it's less magnetic. It's not quite as large of a molecule. And so you can detect sort of these changes in the blood oxygen level over time and in conjunction with specific neural like tasks or things that you're doing in the scanner or stuff like that. And so it can show, again, very small sort of like millimeters of like activity. It tries to break the brain down into these very, very small voxels or components that would be able to give us more of a discrete idea in terms of where activity is in the brain. You can watch this YouTube here to see a more sort of a fuller accounting. And you can see here, for example, what it would look like. And so this is a section right here, and we're looking primarily at this section of the brain. And if you can see here, we're sort of focusing on the back right here towards these occipital lobes. And this is basically looking at the difference in the blood oxygen level, comparing when the light is on or when the light is off. Same cells. So you can see here when the light is off, they're not as active as much, and the MRI signal is lower. But when you turn the light on, then these are light activated cells, and they are more activated, and you can see the corresponding sort of activity saying, okay, here we measure this increase that corresponds with this stimulus, the light turning on. We measure this change. And so then we can say, okay, this region of the brain is more active when the lights are on than when the lights are off. And then you can look to see how it replicates and stuff like this, over time. And so this is then how you can kind of look at this sort of consistent signal to see if it is altering in these regions to test this hypothesis and to show that, okay, this measurement works in looking at changes in oxygen blood flow. So you can take this here and see how these changes are, and then track it over time. If this wasn't turning on the light, but what if this was holding a specific thing in memory, listening to a particular piece of music, and then what do these changes mean in terms of how the brain works for behavior? So this is what fMRI analyses can tell us. We can also be thinking of this as whether you're trying to remember something or not. So then which regions of the brain might be trying to like maintain a particular piece of information versus not. So we can get all sorts of information with regards to how these various brain regions are activating themselves in response to specific types of information. All right. So we also have diffusion tensor imaging, which also has sort of evolved from MRI. This is a special type of MRI, and that similar to oxygen, you have the oxygen molecule in water. So this is all the blood oxygen levels, all about the hemoglobin, having oxygen on it or not. So the DTI uses those same principles, but it tries to use the same basic technology to detect the flow of water through white matter tracks. Okay. So what this is really important is when you put these sort of one of these tracks, these molecules are sort of very thin, the water molecules, and so they're in particular shapes actually within that are different based upon when molecules are in an image, like in a nerve track versus whether they are not a nerve track. And so, and because it's small, then you actually see these water molecules have like a directional flow, in the sense that this is how information would pass through things like that. What is the direct? So with neuroimaging, for example, we can say, oh, look, here are these white matter tracks and here are these processing and stuff like that. But we can't pull them apart at all. They all just blend together. So diffusion tensor imaging actually lets us do that, pulls them apart by, instead of them all just being this sort of conglomerate of nerve facts, it actually looks at some of the way the directions of the water molecules are in those thin tracks. Then it can detect the direction of those, and it can give us these beautiful images here that we can really see how these are tracks that are moving off the side. These ones are moving like straight up, dorsally. These ones head out like laterally. These are your medial sort of density. And you can see here, this is the most dense, and it travels, this is your corpus callosum, and then you can see everything sort of moving out into both of your hemispheres. You can see other aspects of this right here. This is sort of an older image, but these are all the white matter tracks as you would move through different sections of the brain. Slicing through your eyeballs and stuff. So here, as you can see, water can move relatively freely along the axon, but less freely across cell membranes. And so it looks for the shapes of these. And, excuse me. And the direction of this water movement is detected by a coil, and interpreted by a computer. So this is all mathematically brought into place. Look, we have these little electrical fields. They change within these magnetic fields. We can look at, depending upon, we can use all sorts of different like procedures or protocols within the same imaging technology. So the same machine does DTI, fMRI, and MRI. It's just almost different computer programs, basically. Same detection sort of technology. The same thing is used throughout. So the same, machine can go ahead and look at these water molecules shape to determine directionality. It can look at bold in terms of the blood oxygen level, in terms of just doing tasks, and it can do these really nice anatomical images. And it all depends upon what aspect of the sort of oxygenated hemoglobin or any sort of oxygen water molecule with the hydrogen atom, how any of these sort of shift. And then you can determine different types of information from the same signal, but it's just processed differently. The different types of information are required, different types of mathematical sort of techniques to pull the pieces out, but all used by the same machine. And so what the machine can do, direction of the water movement is detected by this coil and interpreted in this mathematical process. And then what it can be used is to delineate abnormalities in neural pathways. And they can also be used to identify changes in fibromyelination, such as the damage that leads to myelin loss and multiple sclerosis. They also have traumatic brain injury and sort of mini traumatic TBI, or just sort of TBIs, which are sort of smaller, that are often associated with more localized regions of trauma or brain damage. But DTI can actually detect if like nerve fibers, nerve pathways are severed, and then which would then be able to say, okay, these are severed, so you'd have to use this type of procedure or this response or this type of treatment, or trying to get these types of, what you could do to help reconnect some of these various regions of the brain to restore plasticity and function. So all of these neuroimaging techniques are used for a wide variety of assessments with regards to our brain and our body's health. All right, and this next one, got a special place for my camera in this slide. Next we have positron emission tomography, and this is what is known as a PET scan. This is used to analyze the metabolic activity of neurons. And so it's good for looking at a brain disease and the working brain. It is also expensive, and a participant has to receive an injection. So the way PET works is an injected radioactive isotope is coupled with a molecule like oxygen, glucose, or other chemicals, things that would be absorbed by neurons for use. They would take glucose in terms of making energy for ATP. They would have oxygen for their function, or any other chemicals that would be absorbed. And PET detects the changes in blood flow, by measuring changes in the uptake in compounds such as oxygen or glucose. So many of the same principles as that active, cells will use up more oxygen and glucose. And then what it kind of we put in this injection that basically tags these cells and says like, okay, here's the cells that we put in at this particular time. They've got this tag. Let's see what happens. In this period of time, for while they're doing this particular task or resting, or we're looking for some measurement of metabolic activity, and so when these molecules, this injection, these are radioactive isotopes, and that doesn't necessarily mean harmful or anything like that. It just means that after a particular period of time, the isotopes sort of like, they break apart. And so what the PET scan can do is it actually detects that sort of splitting. And because when these molecules split into decay in these radioactive components, they do so at actually like 180-degree angles. So that, and you can use that to actually detect what is the amount of metabolic activity that's going on in a particular region of the brain. So when the more oxygen is being used, the more it will bring this radioactive isotope there. The oxygen is being used, the isotope, as it, it starts to decay over time. And then you can see, oh, it's breaking apart and it's being used in this occipital lobe right here, where you have some of this space going on, sort of the lateral frontal lobes, potentially some elements here just which is going on with the temporal lobes period. Maybe this is actually the anterior sort of front portion of the temporal lobes. And so you can see here, wherever this occurs, these red lines, these sort of like opposite molecule over here, these sort of photons, they break apart. In this whole space is a detector. So when these all sort of scatter out in all these different directions, it knows that every single molecule that sort of breaks apart into these sort of protons is going to head in these opposite directions. So if the detector gets one of them and say, oh, I've got one on this side, but it doesn't get the corresponding one, then it knows that it was absorbed by tissue. And so what it will do is it will then use this sort of combination of, I've detected this one, and it's not over here, so there must be some tissue over there. But then if it gets both of these, it's sort of split out the 180-degree angles, then it knows that it wasn't absorbed. So we'll take all of this information and process it mathematically and computationally, and it will give us out this sort of metabolic output right here. And so this actually walks you through. You can see a little bit of how these molecules sort of like head out in these 180-degree angles. This would be an example of one that is absorbed, right in this particular region. And so this is how they're basically detected. So when these protons or positrons are released, they'll collide with these other electrons in the brain. That's when they get sort of absorbed. And when they, do, then they're detected as sort of like absorbed. And when they don't, they exit the brain, just as sort of described here. So this is a little description that walks through the particular pieces. Okay, so that's PET scans. So now we have the magnetoencephalography. I gotta try saying that 3 times fast. It takes some time and practice. So magnetoencephalography, you'll be getting some themes from all of these imaging techniques. This is neural activity that we can detect by generating an electrical field. And this electrical field also produces this small magnetic field. And so this is what I've talked about with a couple of before, that again, neuronal activity, all of these neurons being active, it generates this sort of little electrical field right here. These electrical fields, particularly if there are enough of them, enough of electrical fields, create a magnetic field. And then the magnetic field produced by these, action potentials and these, these individual neurons is really small. But when you put them all together, and there's many of them, and then you can have many action potentials, this actually produces sufficiently strong magnetic fields to be detected. And so this is also like a kind of a detector that would sort of go around your head and detecting these magnetic waves. And what it is it's very precise in terms of determining the location of activity, because these magnetic waves are very much in line with sort of the direction of the neurons and everything like that. And so it can break it down into identifying these specific fields that correspond to specific groups of neurons activating. And so it can get us a pretty high spatial resolution, and it records in real time, but it can also be a fair amount expensive. And this is I believe this is our final slide, and I didn't think this blocked things like this. So, okay, so our final bit of brain stimulation, our final bit of slide, and actually let me check this to see. Oh, nope, we got one more after this. See, two more, and then here Q terms. Okay, so we're almost there. So transcranial magnetic stimulation, this is known as TMS. You can see some of it here, and this uses electromagnetic fields to stimulate or depress activity of the cortex. This primarily, again, works for stuff on the surface. And so you can see here what aspects of it, you put this sort of magnetic, field right here to stimulate, you can see what happens. You can also try to sort of, shut off a particular area to boost function as other areas here. And these can be used in certain therapeutic techniques. And so the magnetic field can basically kind of like, if you put this on here and just line with electrical, it kind of can, hesitate to say sort of like scramble the neurons for a second, but can kind of stop them engaging in sort of synchronous fire. This is only temporary because it's just like bringing up a magnetic field and then moving it away. So it's not going to have any longstanding effects because you're just basically, detecting this magnetic field and then not. And then once you detect these magnetic fields, then you can also adjust these magnetic fields up here to try to have an effect on those particular regions for certain therapeutic techniques. For transcranial direct current stimulation, I think they used to call it TV, yeah. I feel like it used to have a slightly different name, but now they call it transcranial direct current stimulation. And this is basically low frequency electrical stimulation. It's considered non-invasive, and it's used as a possible treatment for depression. And so what it does is that you basically have these particular pieces on your scalp or a particular area. There's a couple of ways of doing it, and it'll create this sort of electrical charge, and this electrical charge will temporarily disrupt the particular area of neurons around that space. And so this can be done for all sorts of testing. You can use it to detect language center to see if you were to go ahead and put sort of an electrical charge here, sort of does it detect your speaking, or, sort of impact your speaking or any of the way that you are choosing your words. They often can focus these on particular areas of the frontal lobe as a target for depression. And because there's this idea that, like depression, these things are sort of like dysregulation. So if you can this idea of that some of these areas are becoming dysregulated. So it's like, well, if we can what if every now and then we kind of try to flush this electrical system sort of clean, so to speak. And but what it does is that these small sort of current, stimulations at these particular points can actually help, regions of the brain that have been less active. They can be more active as well. So this is how it can kind of be treatment for depression. And it can kind of just try to kind of realign certain associations. And that then it can also boost attention and other cognitive functions. It's been a bit experimental in some respects, but it's shifted. That was really many years. And so it's really shifted into something that can be used very effectively in specific situations. And then brain stimulation. So deep brain stimulation is, it may sound sort of scary, but it has like amazing results. And so what this is that you can see here, it's this little sort of apparatus, and it goes through here, and they actually put an electrode in your brain. And this pulse that is generated by here is designed to then sort of stimulate this particular brain region. And so you can see here, this is an example of how it might be used in depression. So this, which of course is far away, it comes in here. You have electrode that sits right in here in this particular region of the brain. It can be different depending upon the type of treatment. But this here this is one for depression. It would sit right in parts of the thalamus and the subthalamic nucleus of the substantia nigra. And that this then, like causing this sort of stimulation can then increase the dopamine released in the substantia nigra. And this basically serves to resynchronize the electrical activity of the subthalamic nucleus. So when they're dysregulated, so if this comes in and gives more regular sort of stimulation, then it can kind of try to synchronize some of these this electrical activity that has been unsynchronized. And it has you have to do a specific type of surgery to place the electrodes. It's used for treatment-resistant major depression. So, and this is stuff where like nothing else works. And it may also be used for Parkinson's, but it was experimental for a number of years, but it's been around for quite a while. And it is, it's invasive. So it's not something that this is not on the first of a medication list or anything like that. But if you have treatment-resistant depression, then what people is they kind of describe it as like, I just can't see out of this hole or get out of this hole. But then the DBS can kind of like lift them up to be like, oh, I can actually see where the edge of the hole is, and then they can use it to help sort of get them out through therapy or other types of medications. This is not like a cure or anything like that, but it's just really fascinating to think how this would work and how it can help kind of resynchronize or restabilize these electro sort of chemical communication pathways. And then our last slide, we're looking at biofeedback. And biofeedback is a way to control autonomic responses, muscle activity, and brainwaves that are not consciously controllable by trying to actually make them a little bit more conscious. So heart rate, skin temperature, muscle activity, and brainwaves can be monitored in biofeedback. And the devices record this physiological activity and signal when intended changes occur. And so this can basically be an associative learning or sort of reinforcement to allow for more conscious directed changes. And so you watch, you're basically engaging in a task. You can see here that there's electrical activity, everything is being recorded. And then, and all of this measurement is basically being displayed here for signal processing. And a patient would actually observe this or could see parts of it. And they would look at sort of the task and they would say, okay, here, and oftentimes a good example of it is actually just doing like a controlled breathing, like balloon task, where you have to kind of like do, like, okay, do this to like blow up the balloon as deeply as possible, and then, not. And so then you can try to get the, and the balloon might not, it's not one that you in front of you, it's like one that's on this kind of computer screen. And then by trying to learn how to shift, an aspect of like maybe every time you actually control your breathing properly, sort of taking a deep breath, then it inflates. And then you actually have to do it to a specific, sort of amount of time. So if it doesn't inflate enough, then it doesn't, you don't get points. And so you basically learn through feedback of what the visual screen is sort of telling you about your performance, and then what you can see from your biological feedback to try to align them. And so it can help you, like, if you see how your body is responding to something, then you can be like, okay, I see this spider over here and I see myself reacting to that. Now I'm going to try to engage in this sort of calming, and then I can see my physiological arousal going down in that moment, then someone's going to be much better able to actually like learn to control themselves, because they're getting feedback in the moment that like, oh, look, you are actually, you know, like biofeedback to confirm that you might be addressing your breath and stuff like that. So as you can see here, it's good for relaxation, reducing headaches, and regaining control of muscles. And it's also really good for brain and computer interface control, things like that. And then now we are on to key terms. There's not that many this chapter. And this was a longer sort of portion of the chapter, but this is the last of it. So we've only had two lectures, two recordings for this. Thank you guys. Sorry about the delay, and have a great time studying this chapter in preparation for the exam.